All right, hello. Thank you all so much for joining us today for this BitBio Discovery webinar. My name is Renee Suleiman, and I'll be the moderator today. In this webinar, Sejla Salic-Hainzl will be discussing how CRISPR-Cas9 gene editing of human-induced pluripotent stem cells can be used to identify drug targets, model disease mechanisms, and study therapeutic responses. Sejla is the Vice President of Research and Development at BitBio Discovery, where she leads a team of researchers striving to push the boundaries of scientific exploration and drug discovery. And with that, I'll turn the time over to Sejla.
Hello. Thanks for a kind introduction, Renee. Welcome to our webinar on how we're combining human iPSCs and human iPS-derived cells and the CRISPR screening technology to sort of help in the development process of new generations of medicines. So I would like to start explaining first maybe the process of how the drug discovery pipeline and the drug development pipeline currently work. So it is a quite lengthy process of approximately, I would say, 15 to 20 years from identifying a target or observing a specific phenotype in a cell to actually delivering a drug on the market. Usually, drug discovery screens start with large screens of small molecules, and while moving on from one phase to the other, the space or the chemical space gets reduced, and usually only about 5 drugs get carried on to clinical trials, and one makes it to the FDA approval and the market. And what researchers working in this space have identified over time is that genetically validated targets are 2 times more likely to be successful in clinical development. And now, considering the time that gets spent to develop a drug for a specific disease, but also the costs that are associated with it, it is quite important to identify already at very early stages if a drug target is a good target and will be successful in the clinics. So this is just some graphics from review where in the year 2022, a total of 50 FDA drugs were approved. And when we look into the human targets, it's approximately 42 that were associated as a human target, and 33 of those drugs had genetic evidence. So it is quite important to know how we can actually genetically link drug targets to diseases. So what the community has done over the past years is different methods, and I would like to briefly talk about each of them. So on the one hand, we can genetically link drug targets by looking into genome-wide association studies. So what happens there is that we look into the genomes of healthy but also diseased individuals, and we try to pinpoint mutations in specific genes to certain diseases. Of course, this whole process can be now quite sped up using artificial intelligence and machine learning platforms. But all of that is sort of in the target identification space. And before it actually gets moved forward to clinics, we do need a way to actually validate those hits that come out of this type of approaches. So one way to do it is also to do more of a hypothesis generated research. This is sort of a one target at a time approach. It is quite time consuming, and the progress is of course very slow. So at BitBio Discovery, we believe strongly in CRISPR screening workflows because they sort of have this power of being unbiased. They can, of course, be done in a pooled or arrayed screening format, and they have a quick turnaround time delivering conclusive data. CRISPR screens, and I'll introduce what CRISPR is and how it can be used in a screening format is that on the one hand, it can be used for target validation, but at the same time, we do believe that CRISPR screens can be used for target identification. So the very early stages of identifying a target that gets moved forward into a drug discovery pipeline. So what is CRISPR-Cas9? CRISPR-Cas9 is actually a DNA endonuclease that was discovered in the immune system of bacteria. What it can do is actually load guide RNAs, which are complementary to a specific sequence in the genome, and they can introduce a double-strand break in the DNA. Once the double-strand break happens, the repair mechanisms of a cell get activated. And most of the time, these repair pathways are sort of the error-prone pathways where different ligases try to stick together the two ends of the DNA. And as I mentioned, it's an error-prone process. So what we usually see is that we have indel formations. So there is specific insertions or deletions in the DNA which cause a frame of out of the frame mutation, which cause a genetic knockout. On the other hand, we can also provide template DNA where we rather generate knock-ins, so we can specify which mutation we want to incorporate into the DNA. Experimentally, especially if we look at the one target at a time approach, we usually have Cas9 expressing cells. We deliver guide RNAs for genes that we're interested to study in. And by this, we obtain cells with a specific gene knockout. We can apply it in different readouts. It can be microscopy, it can be cell viability. And with this, we can see if a specific gene knockout causes a disease phenotype, for example. Now, whatever can be done as a single gene knockout at a time can be also multiplied. And this is sort of more what is done in arrayed CRISPR screens. So there we're working with 96 or 384 well plates. And with this, we can, of course, use automation to process very large numbers of knockouts and cells. But what we like to do at BitBio Discovery is actually pooled CRISPR screens. So while the arrayed CRISPR screens have the advantage that they can be combined with readouts like microscopy, they're actually quite labor intense and require, if they are used in a high throughput, automation and expensive equipment. Now, there are many ways to perform pooled single-cell CRISPR screens, and the technology we've set on is actually crop-seq screens. The advantage of crop-seq screens or single-cell CRISPR screens in general is that it combines scalability with a quite information-rich readout, which in our case is single-cell RNA-seq. So these screens are not done in this 96 well plates that I've shown you before, but they actually can be done in large dishes. Large amount of cells can be cultured. And what we do in this crop-seq screens is that we use lentiviruses to deliver guide RNAs. So we have a step where we do pooled cloning of these guide libraries, and we use a quite low multiplicity of infection to deliver the guide RNA. So by using as little virus as possible, we can actually guarantee that we have one infection per cell. By applying then single cell readout, we can actually read out the transcriptome of every single cell, as well as the guide information. So which guide was delivered into the cell, which knockout caused which transcriptomic phenotype. This is usually accompanied by bioinformatic analysis, where we can see on these UMAP plots cells that have a similar phenotype, which is usually caused by a specific gene knockout. They tend to cluster together on these plots. So CRISPR screens, they're nothing new to the field. They have been used in academic labs, but also in company settings. But what we do believe is very important with CRISPR screens is the model systems we're using. In In the past, immortalized cells were used heavily, and there are certain advantages associated to the cells, simply that they're very robust and scalable, usually very low cost associated with it. But the downside is their physiological relevance. Now, human stem cells have certain advantages. This is usually when we study, for example, developmental diseases. They proliferate well. They are physiologically relevant, depending on the context they apply to, but usually not very informative, because diseases, they usually develop in somatic cells. Primary cells are a good option when studying when wanting to work with physiologically relevant models, but they usually are not very scalable. And the truth is that not every cell type is easily accessible. So primary cells work very well for immune cells. But when we think of neural cells of the brain, it's quite difficult to access those cells. So what the pharmaceutical industry, but also the academic community is trying to currently establish is organoid system or iPS-derived cells. So while their physiological relevance is very high, of course, the complexity increases. They're usually quite expensive models. They are not very scalable. The robustness is also something that works for certain cell types, but not for other. And it's usually not easy to develop these protocols. It doesn't matter if it's certain cell types in a 2D culture, but it gets even more difficult with organoids. Now, what I would like to introduce is the OptioX technology. So as our name already says, so we are BitBio Discovery, and we're using the OptioX technology, which was developed by BitBio, to forward program iPS cells into certain cell types. So iPS cells are pluripotent cells, and they have the capacity to differentiate into any cell type of the human body. And stem cell researchers have done this successfully in the past by doing directed differentiation. What does directed differentiation mean? This is usually applying protocols, so cytokine cocktails, which mimic the development of a cell type in a dish. Certain protocols have been quite successful, but what these protocols usually suffer from is that one usually obtains a mixture of different cell types in the well. So we have quite low purity. The scalability is also usually an issue. So if we think of profiling many drugs or performing large-scale CRISPR screens, the scalability, of course, becomes a big topic. And then usually also the very long culture time. So certain protocols take up to 60, 80 days, or even months. So the OptioX technology is based on overexpression of transcription factors. So for certain cell types, it's quite known that there are transcription factors driving the development of the cells. And these transcription factors get inserted into a safe harbor locus under the control of a doxycycline-inducible promoter. So by applying doxycycline to the cells, the expression of these transcription factors gets activated, and cells sort of get forced to take a new identity. What we've seen with this technology is that we can actually solve all the problems of directed differentiation protocols. So we do obtain precise cell types, high purity cultures. It's a very scalable protocols, and there is also, of course, short culture time. So most of the protocols take 7 to 10 days. Now, BitBio is offering a portfolio of certain wild-type cells. They're mostly in the CNS space, but more of different cell types are currently being developed. And at the same time, the wild-type cells are accompanied by disease models. As you can see, most of those are in the CNS space. So the data that I will be showing you today are done in the cells. Now, talking about CRISPR screens and about the relevance of the physiological model in which these CRISPR screens are performed, we try to develop exactly those pipelines. So we're trying to use the Optio-X cells and engineer Cas9 or other modalities of the CRISPR toolbox into those cells. For this, we are also making use of genetic safe harbor loci, and we insert Cas9 expression cassette. Now, I should point out that Cas9 in this case is constitutively expressed. So we have to do quite some extensive QC to make sure that the Cas9 expression is not doing something weird to the cells. So on the one hand, constitutive Cas9 expression could hamper the forward programming potential of the cells. So as a first step, we are taking the wild type cells, so the Cas9 non-expressing cells and the Cas9 expressing cells, and we simply perform the forward programming differentiation. We assess the differentiation efficiencies, we check for expression of certain cognate marker genes, and we usually also check on the transcriptomic level if iPS cells, but then also the more mature cells are behaving in a similar way. Now, during forward programming, silencing could occur. So we have to make sure that Cas9 is expressed in the forward programmed cells. And then what gets even more difficult, in order to do that, we have to find a way how to deliver the guide RNAs into those cells. I will show you now data following exactly that workflow for the glutamatergic neurons, and then later for microglia. So we took the iPS line of the glutamatergic neurons harboring the transcription factors driving the forward programming process, and we forward programmed the wild type cells, the Cas9 expressing cells, and by showing you this IF, I hope I can convince you that actually Cas9 expression is not hampering the forward programming potential. And by showing you this PCA plot of transcriptomic, on the transcriptomic level, that at the iPS stage, we see minimal variance at neurons day 4, day 18. There is very little difference between the Cas9 and non-Cas9 expressing cells. The next step is, of course, to bring in a guide RNA to see if Cas9 is actually functional and expressed in those cells. So we are taking usually the cryopreserved material from BitBio, where upon thawing, cells reach very quickly within the first 7 to 11 days a mature phenotype. And we're delivering the guide RNA at different stages to see how long is Cas9 expressed in those cells. We have 2 different approaches. One of them is a lentiviral delivery. The other is delivery of synthetic guide RNAs. And then we assess the knockout efficiency on the DNA level, but also on the protein level. So this is some of the data. So we, upon revival of the cells, transduced cells at day 11, at day 1 and day 3. And in this case, we're targeting a gene called SOX11. So we designed a guide RNA targeting exon 1 of SOX11. And by performing amplicon-seq, it means we're just amplifying those regions by PCR and submitting it to next generation sequencing. We actually can see the percentage of indel formation. So how many of the sequences are still wild type and how many of them have an insertion or a deletion. As you can see, we use a lipid-based approach and lentiviral delivery. And the results that we obtain and the efficiencies that we obtain are actually quite comparable. Of course, we used a non-targeting guide, so not targeting any sequence in the genome as a control. Now, what is even more important as a readout is that we can confirm that we see the knockout efficiencies confirmed also on the protein level. So in orange, we have the non-targeting guide. In blue is the DAPI staining. So it's the nucleus of the neurons. In red, we have the expression of SOX11. And then when we overlay the nucleus and the SOX11 signal, we get sort of this purple signal. So now I think it's quite convincing, and you can already see it by eye, that once we use this guide RNA targeting SOX11, we do see that the knockout is quite efficient. So the cells are not expressing, or only very few of them are still expressing SOX11. So now using image analysis tools, we can quite nicely identify the knockout efficiency. So first is the no virus control. Then we have a non-targeting guide RNA. So in both cases, we have almost 100% of SOX11 expression. And then once we deliver the guide to the cells, we do see that only 5 to 10% of the cells still express SOX11. Now, having this physiologically relevant model, seeing that it actually can express Cas9 and that we can deliver guide RNAs, generate knockouts, the next obvious step for us is to build a single-cell CRISPR screening workflow. I'll briefly would like to touch on how those CRISPR screens with iPSCs have been done before, and why we think that it actually matters to introduce CRISPR perturbations already in differentiated or in forward programmed cells. So for a long time, researchers have been generating Cas9 expressing iPSCs, but the difficulty was, of course, to deliver the guide RNA in the differentiated cells, or even to scale those protocols so that many guides can deliver it at the same time. So usually, the guide would be delivered in the iPS stage, and then cells would be differentiated to study sort of certain biological process. The difficulty with such an approach is that knockouts, of course, have also an effect on pluripotency. Specific genes might be essential in iPSCs, but not in neurons. And then they could also be doing something to the differentiation process of the cell. So a rather simple readout is sort of just assessing the survival of the cells. So is any of the gene knockouts having an effect on the survival of the cells? And with this plot, we can see quite nicely that we identify certain hits that are doing something to the cell fitness. But it's very difficult to say if this is actually biology that is meaningful for in a neuron, or if this is actually affecting any of the other processes that I mentioned. So when it comes to screens for neurodevelopmental disorders, this might still be a good approach. But if you end up with a list of, let's say, 200 hits and you have to validate each one of them, it would be quite good to know which process you're actually validating. Now, we do think that timing is actually key. So it's important to understand the biology that is being studied with a CRISPR screen. And what we try to develop is really a CRISPR screening platform in which the biology of neurons or microglia, sensory neurons, or any other somatic cell type can be studied. And to showcase why timing is important, we actually performed exactly such a screen. So on the one hand, we delivered the guide RNA in the iPS stage and then forward programmed cells into glutamatergic neurons. And in the other screen, we delivered guide RNAs into the forward programmed cells. So what we're studying here is actually which knockouts are lethal to the cell. And what you can see quite nicely in this plot is on the y-axis, we're plotting the knockouts that are introduced in the glutamatergic neurons. On the x-axis, the knockouts that are introduced in the iPSCs before forward programming. And what we can see is that certain gene knockouts like HRS1, CARS1 are quite lethal when introduced in the iPS stage and then forward programmed, whereas there's not much happening in the neurons, so they do not seem to be essential in neurons. Now, why I'm pointing out exactly these two genes is that on top of doing a survival-based readout, we also performed single-cell RNA-seq. So with single RNA-seq, we can capture the transcriptome of every single cell. And while if we would have performed the screen by delivering the guide in the iPS stage, we would have never been actually able to capture the biology of these genes upon knockout in glutamatergic neurons. So our main hits from the screen that we performed, and you can see quite nicely, they belong to the same protein family. On top of that, we've also identified VCP and ELP3. And all these genes are actually associated with the Charcot-Marie-Tooth disease, which is a neurological disorder. When diving deeper into the pathways that are affected in the cells, so simply looking into the differentially expressed genes, we did identify on the one hand the activation of the unfolded protein response, which is a quite good biological control because these proteins are catalyzing the attachment of the respective amino acid to the tRNA, which of course has a function on protein synthesis, but also protein structure. And once this pathway is disturbed, so the folding of the proteins, the unfolded protein response gets triggered. At the same time, we did identify quite some processes which are associated with lipid metabolics, cholesterol biosynthesis. And when looking into literature, we did also identify that we're not the first one seeing that, but that actually this lipid metabolisms are even considered as a potential therapeutic strategy in curing this neuropathy. And when looking into clinical trials, if any of them have been started, I did manage to find one that was started in 2020. So with this, we can actually show that the CRISPR screening workflow that we've developed is actually quite useful for target ID, but also target validation, and that by performing these genetic knockouts, we can actually study the biology of neurons. Now I would like to switch to a different cell type, which is also a forward programming protocol powered by OptioX. So microglia, they're immune cells of the central nervous system, and they come sort of in different flavors. So immune cells, and especially microglia, they have different activation states. I think that this figure we already know by now is a quite simplified view of this activation states. And on the one hand, they can be activated to be pro-inflammatory, so to clear, for example, degenerating neurons, and they secrete cytokines which have a neurotoxic function. But on the other hand, they can also be activated into a state that is anti-inflammatory, so they have a neuroprotective function. On the BitBio homepage, there is a lot of information on the microglia and also functional readouts. I'm showing just one here, which is one of the function of the microglia is that they can phagocytose. So they actually can clear the degenerating neurons. We are showing this here with an assay that is based on providing sort of E. coli particles that are coupled to a fluorescent marker. And once those particles get phagocytosed, we can see an increase of the fluorescent signal. So this is a time course of experiment of about 24 hours. And we do also see by providing an inhibitor, we can inhibit the phagocytosing process, and we do not see an increase in the red signal. This is just a quantification. This has been also tried with yeast particles, and we do see that the microglia that we are working with are actually functional. Now, what I've showed you for the neurons, we've had to go through the same process of developing these Cas9 expressing lines in order to use them for CRISPR screens. I'll cut that process now a bit shorter because I introduced you already to that. So in this case, we're inserting a Cas9 expression cassette into a safe harbor locus, and we're characterizing the cells. So on the one hand, on the transcriptomic level, we see very little differences between the Cas9 expressing and non-expressing cells. But at the same time, we also can confirm that the forward programming potential is not hampered in these cells. This we've also assessed with a functional assay. So here we're using the non-expressing cells and the Cas9 expressing cells, and we're treating them with LPS. LPS is actually a component of the outer membrane of gram-negative bacteria, and it causes microglia to activate, and this we can measure simply by cytokine secretion. So both cell lines are can be activated, and we do also see that their secretion profiles are quite comparable. We've also mastered the protocol on how to deliver guide RNAs into the cells. For now, the protocol on a synthetic guide delivery is still under development. But what we can see quite nicely is that we achieve high knockout efficiencies. And while with the glutamatergic neurons, we have a quite narrow window until when we can deliver guide RNAs, this is not the case for microglia. So it seems that microglia, even in later stages, can still express Cas9, and Cas9 is functional in those cells. This is just a time course experiment to see how long Cas9 is functional. Now, in a next step, we aimed again in setting up CRISPR screening workflow. This is sort of a proof of concept screen to see if these cells can be used for the same pipeline. In this case, we have two conditions. On the one case, we have microglia that are not activated, so they are not treated with LPS. And then we have culture that gets LPS treated after the knockouts get introduced into the cell. What we included as a control to simply see if this workflow works, we have included the toll-like receptor 4 pathway, which is sort of the pathway responsible for the activation of pro-inflammatory cytokines upon LPS stimulation. So TL4 is the receptor to which LPS binds. Now, before I go into the outcome of this screen, I would like to introduce you to targeted sequencing. So targeted sequencing is an approach we like to use because when looking across different screens, we usually see that upon a perturbation, there are not many genes being differentially expressed in a cell. So usually the majority of proteins in a cell stay unaffected, and there is only a few of them that get differentially expressed. So we did not come ourselves up with this method. This method was developed by Daniel Scheifogel. It's the TAP-seq approach, but we do have ways to design primer panels which are specifically enriching only for these genes. So what it's important to know before starting such a readout is the phenotype that you want to understand or that you want to capture in your screen. The advantages of targeted single-cell RNA-seq is that whole transcriptome readouts are usually quite expensive when it comes to the NGS costs. So we need approximately 50,000 reads per cell, whereas if we're using a more targeted approach, we can reduce the costs approximately by 5-fold. So this is what we did for the microglia. We did know that we have two conditions. One is the resting microglia, the other is the activated microglia. And by performing bulk RNA sequencing, we were able to identify a transcriptomic activation signature. So we identified approximately 1,610 differentially expressed genes. And by using bioinformatic analysis, we could narrow down that activation signature to 258 genes. This is just a plot showing that the targeted sequencing also enhances detection of mRNAs in a cell. So while using whole transcriptome sequencing, let's say approximately 5% of the cells would express a specific mRNA, or we would be able to capture that mRNA. And by using the targeted approach, we do see approximately 25 to 30% of those cells cells expressing this mRNA, which of course gives us a greater depth when looking into our data. We performed the screen and used cosine similarity analysis to uncover modulators of the activation state of the microglia. So this means that our activation signature, if you want to depict that on this on this color wheel, we would have sort of a value for a resting state and for an activated state, and we would try to understand which of a knockout is causing our cells, causing cells from an activated state to switch to a more resting state. And by simply taking the angle between those vectors, we can calculate the cosine of this angle. And with this, it gives us the percentage of the similarity, for example, between vector A and vector B. This is how our hits look like. So the screen was performed. We perturbed approximately 110 genes, had 2 different conditions, plus minus LPS, performed single cell sequencing, and used cosine similarity to analyze the data. So what we're plotting here is exactly this activation signature. So we have seen genes which are upregulated and downregulated when comparing the non-targeting cells, LPS stimulated, and non-targeting cells without LPS stimulation. And we do identify hits, and what we like to see is especially the hits that are highlighted in orange, because those are the genes involved in the toll-like receptor 4 pathway signaling, which gives us confidence that we actually can find biologically relevant data in our screen. So all of these hits can now, of course, be further validated in a more arrayed approach. So on the one hand, we have functional assays like the cytokine secretion assay, but also the phagocytosis assay. Now, as I mentioned before, BitBio is offering the wild type cells, but also the diseased cells. So the CRISPR screening workflows that we've established can be used to study sort of the mechanisms involved in diseases by performing one and the same screen in the healthy cells and then one and the same screen in the diseased cells, and trying to see how specific gene knockouts are modulating the effects observed in the diseased cells. While the IO glutamatergic neurons will be launched as an early access product next week, we're currently developing the microglia and working on all the other cell types, because we do understand that there might be also a need for such cell types on the market. So we're having this in-house pipeline to perform these large-scale screens, but at the same time, together with BitBio, we're offering the cells as a product. Just as an outlook, so I talked now a lot about CRISPR knockout screens, so where we're really knocking out genes, but the CRISPR toolbox has much more to offer. I'm just mentioning here CRISPR activation. So in the past, we've also been quite successful in performing genes where we are activating genes that are not expressed in cells. For that, this quite colorful UMAP is showing that there's a depiction of 221 genes, and cells are colored by the guide they're expressing. And we do see that in this screen, at least, the activation has caused quite some phenotypes that we're looking further into. And at the same time, of course, CRISPR interference screens can be performed where we're not fully knocking out genes, but just repressing their expression. Here, we've started developing the cells. So we just tested 2 different cell surface markers where we do have cells in gray expressing those surface marker genes. And then upon delivering the guide with a lentivirus at different concentrations, we do actually see silencing of those 2 cell surface markers. And with that, I've actually come to an end. I hope that I could give you a nice introduction into how CRISPR-Cas9 can be used in drug target validation, but also identification, that we've developed a crop-seq platform where we can do CRISPR perturbations with single-cell transcriptomic readouts. We do believe that transcriptomic represents a very generic readout, which is applicable across many areas of biology. I've walked you through the development of our CRISPR-ready IO cells. So showing that in the glutamatergic neurons, we identify genes which are associated with the Charcot-Marie Tooth disease, and that we've performed a targeted crop-seq screen to identify modulators of microglial activation. And that in general, targeted sequencing offers a good readout with a greater dynamic range and lower NGS costs associated with it. With that, I would of course also like to thank the whole BitBio discovery team and also the BitBio colleagues, and of course you for your attention, and I'm happy to take any questions you have.
All right. Thank you so much, Sejla, for that great presentation. Now we are going to go through some questions that we've received. So our first question is, do you observe silencing of Cas9 in these forward programmed cells?
Yes, so we do think, and I've shown you some of the data, so in the glutamatergic neurons, we clearly see that around day 4 post-revival of the forward programmed cells, we do see that Cas9 expression goes down. That's why we usually deliver guides only until day 3. And we do see that we can perform a CRISPR screen with it. So it's not just a portion of cells which still express the Cas9 and the other not. And in the microglia, we haven't seen any silencing. So we do have to see how this holds true for the other cell types that are currently being developed.
Okay. And another question we have here is, what is the potential scale of these single cell CRISPR screens? So do you use genome-wide libraries or only focused libraries?
Yeah, so both screens that I've shown you for the glutamatergic neurons, but also the microglia are proof of concept screens. So they're rather small screens. It's approximately 100 genes that we're targeting. So the screens can be expanded. The difficulty with these cells, of course, is that they're not proliferating. And also another difficulty is that cells need to be selected. So by going in with a low MOI, we have only approximately 10% of the cells that we have in the dish transduced, so harboring a guide, and with that a knockout. So I would say the limiting factor is a bit incubator space and also the selection protocols, but we're working on scaling the protocols.
Okay. And another question is, do you think that there are any inherent limitations to which cell types could potentially be generated by your OptioX cell reprogramming technology?
Yes, so there are large discovery programs, I think, ongoing, not just at BitBio, but also in academic labs, to find the right transcription factor combinations. Theoretically, we do think that there are no limits, but it is quite an effort to identify which transcription factor combinations need to be inserted in safe harbor loci to obtain any cell type of the human body. But we do believe very strongly that this is possible.
Okay, that's great to hear. And an additional question we have is, what do you see is the purity of these forward programmed cells? Do you ever observe any contaminating populations?
Yes, so we do know that in the glutamatergic neurons, and I don't know if attention was played to the UMAPs that I've shown showing the single cells, we do see a population of cholinergic neurons. However, it is somewhere between 10 to 15% in our hands, and that is quite reproducible. So with every CRISPR screen that we've done in the glutamatergic neurons, it's not a population that is overgrowing neurons. So it's just a different neuronal subtype.
And for CRISPR-A or CRISPR activation, are you using viral-derived transcriptional activators, or have you looked into naturally occurring human activators?
So in this case, our dCas9, so the catalytically inactive Cas9, is fused for CRISPR-A to Viper, and for CRISPR-I to a CREP domain. So okay. This is currently the configurations we're working with.
And does BitBio Discovery offer any screening services? Can you go into more depth about what your offering is?
Yes, so we are a single cell CRISPR screening company, and we offer this service also to third parties. So currently, the CRISPR ready lines are the two cell types I've shown, but we can have the capacity to turn any of the other currently available Optio-X lines or disease model into a screening line, and the workflows are quite set up to perform these screens.
Great. And how exactly do you select for guide RNA integrations in post-mitotic cells?
Yes, so we can use, so for the neurons, for example, we used puromycin selection. That worked out quite nicely. So we had approximately an MOI of 0.2 before selection, and after selection, we had around 95% of GFP positive cells. I believe that the neurons could also be selected. And this is the approach we went for with the microglia. So we sorted cells before performing the single cell protocol. But we do have to develop sort of selection methods for each of the cell type. There are different routes we're taking.
Mm-hmm. Okay. And finally, you mentioned at the end CRISPR activation and CRISPR interference, along with just the conventional CRISPR knockout system. How often do you use CRISPR-I and CRISPR-A in comparison to conventional CRISPR?
Yes, so CRISPR-I, we haven't used yet in a screening format, but CRISPR-A, we've used quite often in-house. So with CRISPR-A, the crucial part is actually the guide RNA design. So this is what makes these workflows a bit more tricky, because the guide RNA design is not as well understood as for Cas9. But we have an in-house bioinformatics team, which is very good in developing guide RNA design tools. And we're using all the data that we're generating in-house to refine those tools. So we are sort of getting better with every screen.
Great. And do you observe any silencing of Cas9 in forward programmed cells?
Yeah, so that goes along the very first question. We've seen that in glutamatergic neurons, for now, not in microglia, and with the other cell types, it remains to be seen.
Okay. And how exactly are you planning on following up on lipid metabolism as a therapeutic strategy in CMT disease?
Yeah, so we ourselves are not a drug discovery company. So we are usually working together with third parties. And this is exactly the companies that are working more on drug discovery and drug development. So we can provide our platform to profile small molecules in combination with CRISPR screening. So we ourselves are not following up on that. Yeah.
Great. Okay, well, that is all the time we have for questions. If you have any additional questions, please reach out to info@bitbiodiscovery.com or visit bitbiodiscovery.com to see more information about BitBio's technology and services. Stay tuned for more webinars to come and subscribe to our email list to stay up to date. Thank you again to Sejla, and thank you all for attending today's webinar. Take care.
Thank you.